One. In this mini podcast, I am super jazzed to talk to you about using fat as a fuel and why endurance athletes like to become fat adapted. In this in this podcast, we're going to follow the process by which fat gets broken down in our body. And we're going to think, what is it about fat that makes it endurance mountain sport athletes' favorite fuel? Fat gets stored primarily in our adipose tissue. And guess what? It is very lightweight and it is very anhydrous. What that means in a nutshell is that this molecule that's worth a lot of energy, it weighs very little. So think about how efficient that is. It's like really good backpacking food. It weighs almost nothing, but it gives off so much energy. This is in contrast to glycogen. Remember glycogen, it's the storage molecule made of glucose monomers. It is the carbohydrate storage molecule that we store in our liver and our muscles. Glycogen contrasts with fat. It's very highly hydrated, meaning that it's very heavy. So imagine if you were going backpacking with boiled pasta noodles, very much heavy, right? And maybe not worth the amount of energy that they give you for the amount of weight that you're carrying. So we can think about why it is then that endurance athletes really want to get fat adapted. But let's think about how this fat gets freed from its adipose tissue and transported to the tissues that want to break it down and want to use it. So fat starts off being stored in these adipose tissues. It's all pent up in that tissue and it needs to be enzymatically freed, let loose from those tissues so that it can then get delivered, say, to our slow twitch muscle cells that are highly adapted to using it. Remember, they're really good at aerobic respiration and can really use fat as fuel. So inside of the adipose tissue, enzymes set the fats free, and then a protein called albumin carries them through the blood and to the slow twitch muscle tissue, where they then can get broken down. And we'll look at that at a molecular level shortly. But first, let's think about just zooming in to a slow twitch muscle cell and going inside of one cell and seeing what happens when a fat gets dropped off in the cytosol of that cell. So let's resituate ourselves in this cell. Here, I'm showing a fatty acid, what's called an FFA or a free fatty acid, inside of the cytosol of a cell. Now, it turns out that free fatty acids are not generally found in that form because look at the form that it has. It has a carboxylic acid, if you recognize that from chemistry, and then a long hydrocarbon chain. This looks a lot like a detergent molecule, like your Dawn detergent, and it's going to solubilize membranes. So this is this is like armed and dangerous in this form. So a fatty acid is going to require a bodyguard, a molecule, quote unquote, that has to attach to that free fatty acid and make it safe, but also deliver it to its target destination, which is inside of the mitochondrial matrix. And that is where fats are broken down, is inside of the mitochondrion. So let's look at the technicalities of how this free fatty acid gets escorted by a bodyguard to both make it safe and to also get it in to the mitochondrial matrix. So notice something before we do that. This part of the fat, the C double bonded to an oxygen, and the long hydrocarbon chain, this is known as an acyl group. This acyl group is the way that the fat will get bound to another molecule is through the bond from the carbon to another molecule there. So don't get overwhelmed when you look at this next step. Recognize that we're still situated in the same spot. So let's go back for just a minute. We're situated in the mitochondrial inner membrane space right here. We're zooming in and we're looking at how do we get this across the inner mitochondrial membrane. Remember, it's very exclusive, like the bouncer at the exclusive dance club. We're going to see how does it get across that. So it involves a lot of bodyguard transfers. So we see first that carbon bound to long hydrocarbon chain, that's R equals this long chain. So just recognize that. So this is the fatty acyl group right here attached on to none other than coenzyme A. Remember meeting coenzyme A in the TCA cycle. Coenzyme A is the bodyguard here, attaching to the fatty acyl group, and allowing it to be activated. We call it activated and ready for transport. But the coenzyme A itself is not the right bodyguard to take it into the cell. That correct bodyguard is a molecule called carnitine. So the coenzyme A bodyguard transfers the fatty acyl group onto the carnitine, making a fatty acyl carnitine molecule. That molecule is now given high clearance to get into the mitochondrial matrix. There's a transporter for it that allows it to get in. And so this enzyme that allows for the transfer of the activated fatty acyl group is called carnitine acyltransferase 1. And this enzyme is highly regulated, meaning that if your body doesn't need to use fat, like say you're sitting around eating popcorn and watching a movie, that enzyme will get turned off. But if you're out on a 4-hour mountain run, that enzyme is going to get turned on, right? I get turned on by 4-hour mountain runs too. So it's just like me. And so that will allow the acyl carnitine to take the fatty acid group into the mitochondrial matrix. And then now the coenzyme A bodyguard, that's the primary bodyguard, can pick it up inside of here. It's an internal coenzyme A molecule. And now we get that fatty acyl CoA again. So it's now ready. This molecule is now ready for the fat to get broken down. So the coenzyme A bodyguard is what takes the fatty acyl group into the pathway that breaks down fats. That pathway, it occurs inside of the mitochondrial matrix, and it's called beta oxidation. So beta oxidation is a spiral pathway. It's called a spiral pathway because picture this really long, say 16 carbon long fatty acyl group. Each time the pathway spirals, 2 carbons are removed or broken off of the fat. So it spirals once, 2 carbons are lost. Spirals again, 2 carbons are lost, right? Until the last spiral, when 4 carbons are lost, because there was only 4 to start with, and 2 carbon molecules are formed. So you can think about how many times a beta oxidation would have to spiral if it was a 16 carbon long fat. So the fatty acyl CoA rolls into this spiral pathway in step 1, and that is it is an oxidation step. It forms the high energy molecule FADH2 that we've met before. Remember meeting it in the TCA cycle, where it is worth a lot of power by pump. It contributes to the pumping of protons, the making of the proton motive force, and the making of ATP via oxidative phosphorylation. So energy is generated in step 1. There's an oxidation. The fatty acyl group gets oxidized, forming what's called trans delta 2 enoyl CoA. Some of you will recognize that means that there's a double bond now introduced into the fatty acyl side chain, and that changes the nature of it. Now, in step two, there's a hydration, and that causes the third carbon in the chain to become a hydroxyl group. And then there's another oxidation. I mean, remember the pathway is called beta oxidation. So there's lots of breakdown, lots of oxidation, forms another high energy electron carrier, remember NADH, that can contribute to the proton motive force in the electron transport chain. And this gives us a ketone called ketoacyl CoA. So now the third carbon in the chain is fully oxidized, and at this point, an acetyl CoA is lost. Hey, you recognize acetyl CoA. It's the molecule that goes to the top of the TCA cycle, and it can get broken down in the TCA cycle. So then that generates more NADH, more FADH2, more ATP. And so that acetyl CoA is packed with power as well. Oh my goodness, a fat is packed with power. The FADH2 contributes to energy at the ETC, the NADH contributes to energy at the ETC, the acetyl CoA contributes to energy by entering into the TCA cycle and then contributing to more NADH and more FADH2 to make more ATP. So there is so much energy. This is just one spiral of the pathway. It's going to spiral again and again and again, right? How many times is it going to spiral? If it's 16 carbons long, you can think about that, because remember the last spiral yields 2 acetyl CoAs. So we get a lot of energy coming off of beta oxidation, but that energy can only be formed in an aerobically respiring cell. Why is that? Because that's because of all this reducing power, all of this FADH2, all of this NADH, all of the stuff coming from the TCA cycle. It's only worth energy in a cell that is using the electron transport chain and is got a terminal electron acceptor, oxygen, in order to keep on pumping protons and keep on making ATP using the ATP synthase machine. So for a long time, this super confused scientists and exercise physiologists especially, because they thought that only a little bit of energy could come from fat when an athlete was near their max heart rate, because fat can only be used aerobically. But anecdotally, coaches and others were observing athletes themselves that they could keep using a lot of fat, even at like 88% of their max heart rate. And so what was going on there? Well, this was all about the fact that the slow twitch muscle fibers in a highly trained endurance athlete have become max efficient at aerobic respiration. So the slow twitch muscle fibers, remember, they were forming more mitochondria, more proteins to support aerobic respiration, more capillary oxygen pathways to the cell. That's why we do all of the over distance in the summer, is to maximize our fat adaptation, our capacity to use fat even when we're going fast. We can build up that capacity of our slow twitch muscle cells. So I've done my best to draw a picture of this. Mind you, it is just an artistic rendition. The picture on the left is the one we've looked at, that is the actual, the actual imagery of a slow twitch muscle, in which you're seeing cells that are, or I should say, this is from a muscle. We are seeing slow twitch muscle cells, which are staining dark. Remember the staining the dark cells, meaning there is a lot of mitochondrial density, a lot of protein density, where there's a lot of, say, myoglobin that can pick up the oxygen. There's a lot of capillary feeding of these cells that is, so we have max efficiency when it comes to the use of oxygen, high mitochondrial density. Remember, lots and lots of surface area on the cristae of the mitochondria, lots and lots of components of the electron transport chain, max capacity to efficiently use the oxygen that is delivered to the cell. Then max efficient pickup, right? The concentration of the molecule called myoglobin, the protein called myoglobin that picks up oxygen, it's um it's maximized. There's a lot of myoglobin density, a lot of capacity to pick up oxygen, and a lot of capacity to deliver oxygen. We become, the more and more we train our slow twitch muscle cells, the more and more they become capable of receiving, and the more and more delivery capacity we're asking of our capillary beds. And so we're more and more efficiently utilizing oxygen. So this also is like a hand-in-hand relationship with our fast twitch muscle cells as well. So remember, this image was from a highly aerobically trained endurance athlete, where we're seeing a higher concentration genetically of slow twitch muscle fibers. But also, it's worth noting that those are that even if genetically you have those, training them allows them to become more efficient at their job. And it also allows them to become more efficient at relationships with our fast twitch muscle fibers. And the fast twitch muscle fibers, of course, they're meeting those anaerobic needs really effectively. And of course, creating lots of byproducts from that anaerobic metabolism. And we know that our highly evolved slow twitch muscle cells are also supporting them by removing byproducts of that anaerobic metabolism. So this is why we want, as endurance mountain athletes, to become fat adapted. We want to be able to use mostly fat even at high heart rates, so that we can hold on to our glycogen stores for that very last finishing sprint. We don't want to deplete our glycogen stores too fast. We want to hold on to our glycogen stores, use fat for as long as we possibly can throughout a majority of the race, and hold on to those glycogen stores for that very last finishing sprint. Okay, well, I don't know if it's cool to say that's fat anymore when you mean that's cool, but in this case, I think it's appropriate. So that's fat.